Hi, Michelle Glass here. Welcome back to another episode in our Chapter 28 lecture series. We are here still talking about the female reproductive system, and our job for this video is to discuss oogenesis, that is the formation of the female gamete, which is referred to as the ovum or ova for plural, or of course the egg cell. Now, we still see that the same steps of mitosis and meiosis are occurring, and we have some similar nomenclature for the female that we did in the male, but we have some key differences. Look at this. This process that we're looking at right here is happening in the fetal ovary. So that means when that baby girl is still wrapped up inside her mama's womb. So this is happening 3 to 7 months of fetal development, so before birth. In her little tiny ovaries, you have your stem cell, the oogonia, which is going to undergo mitosis. That's the same as what we saw for males, but it was called the spermatogonia. And you see, of course, at the end of mitosis, you have 2 cells. So you're going to replace that oogonia with a new one. And then you're going to have a cell that's going to go through meiosis. So that's called the primary oocyte. Meiosis 1 actually starts in this little tiny fetal ovary, but it stops at prophase 1. So we start meiosis 1, but we don't complete. And no completion is going to occur until puberty starts up. So that little baby girl is born with all of her primary oocytes frozen in prophase 1. And this thing to keep in mind here is that means that these primary oocytes are as old as she is. So if she's having her babies when she's 20, she's working from a 20-year-old primary oocyte cell. If she is instead having her babies when she's 40, then she's working with a 40-year-old cell. So that's why we start to see some complications occurring as and viability of eggs getting less as women get older. So we do see a definite time frame for women's reproduction, much different than what we saw for men. Now, when she's born, she has all of these, again, primary oocytes frozen in prophase 1, and that is there stopped until puberty. We can refer to this as the primordial egg nest, and she's born with about 2 million of these cells, so clearly way more than she needs. That's okay because atresia occurs. So at about, the onset of puberty, she's down to about 400,000 possible primary oocytes, still way more than she needs. But we'll see that atresia continues throughout her life. Now at puberty, we're going to see the onset of the release of gonadotropin releasing hormone at the hypothalamus, which is going to talk to the adenohypophysis. We know this. We saw this back in the endocrine chapter. We saw it for the male, and we're seeing it again with the female. This is going to signal the release of both FSH, follicle stimulating hormone, and LH, luteinizing hormone. All of these we know from before. We will see that these hormones are actually named based on cells in the female ovary. So our follicle stimulating cell is going to, guess what? Stimulate the follicle cells in the ovary. Now remember, we had this picture of our ovary before, and we see the little egg nest there, and then we see some follicles at different stages. So the follicle cells are going to be those supporting cells wrapping around that primary oocyte, or a secondary oocyte if we're a little bit further along in maturation. We will call these follicles based on the follicle development, and we'll see that the FSH is actually going to help stimulate our mature follicle to actually become what's called a Graafian follicle. Okay, so remember we had our primary oocyte frozen in meiosis 1 and prophase 1 at birth. And then we see with the onset of puberty, we start to get these releases of FSH, follicle stimulating hormone. And this is going to reactivate those primary oocytes to move out of prophase 1 and move into, finishing meiosis 1, resulting in secondary oocyte. Now, in males, we produce 2 secondary spermatocytes, but in females, we only produce 1 secondary oocyte. And notice that it's not an equal division. Basically, we're going to get one cell with all of the cytoplasm but half of the genetic material, and then we're going to get this other teensy tiny cell that's pretty much just a package for half of the genetic material, and we call that the polar body. It will never ever become anything. Ultimately, it's just disintegrated and removed by the body. But this is a way for us to split our genetic information. Then we see that the secondary oocyte is actually the cell that is going to be released at ovulation. It's going to be frozen in metaphase 2. So our secondary oocyte starts meiosis 2, but it doesn't complete it until after fertilization occurs, which means that secondary oocyte, or you can call it an immature ovum, has to travel into the uterine tube, meet up with sperm in order to complete meiosis, in order to become an ovum. At the completion of meiosis 2, you get the formation of a second polar body. Again, you don't get this equal division of cytoplasm. All of the cytoplasm goes to the ovum, and then you get half of your genetic information wrapped up in this polar body, and again, it never ever becomes anything. It does get disintegrated and removed by the body. If you don't have fertilization of that secondary oocyte that was released, it's just going to be disintegrated by the body and not become anything either, and never become a full functioning ovum. So notice when we're looking at oogenesis that because of the formation of polar bodies, the female is only producing one viable gamete with each meiotic event. So only one ovum is going to be produced as a result of that primary oocyte finishing meiosis. Those polar bodies are disintegrated and removed by the body, so they are not ever a viable gamete. Those developing oocytes, remember, are surrounded by those special types of cells called the follicle cells inside that ovary. We have follicles at different stages of development in the ovary, and we start to see that it's with the onset of puberty that those follicles become activated. So we're seeing not just a change in that primary oocyte finishing up meiosis 1, becoming a secondary oocyte, and then moving into metaphase 2, but we also see that the follicle is changing. So we have what's called a primordial follicle. Those are what you're born with. Primary follicle, secondary follicle, tertiary follicle. Now I think about this as an assembly line. So you start to trigger, some of those primordial follicles to become maturing and changing. And that can take about a year. And then once it's a primary follicle, it can take about 4 months to become a secondary follicle. Then it's another 2 to 3 months before it becomes that tertiary follicle. Along the way, some of these follicles are going to degenerate. You're only ever ovulating maybe 500 secondary oocytes out of that collection of like 2 million that you started with at birth. So we're seeing that we're getting little bits of development along the way, but it is not every single one of those primary follicles, thank goodness, becomes an ovum. Those tertiary follicles are maturing and developing in about 28 days, and that is part of our ovarian cycle. So you're going to end up with like a couple of 3 to 5 tertiary follicles, but only one will become a mature Graafian follicle, and it is the mature Graafian follicle that will actually release that secondary oocyte at ovulation. So when we're talking about ovulation, you should just have one mature Graafian follicle releasing a secondary oocyte. So just one secondary oocyte is released with the ovulation, or excuse me, with the ovulation or the ovarian cycle. And then what we see is those follicle cells that remain in the ovary become what's called the corpus luteum. The corpus luteum will be releasing some important hormones, which we'll talk about here in the next video. If no pregnancy occurs, that corpus luteum, after about 12 to 14 days, will become the corpus albicans, which is basically a little piece of scar tissue that does not produce hormones. All right, that's it for now.